before. There's still lots of place in the front row where somebody wants to come. I think we will get started here. So this lecture is actually streamed to the lay people as well. So there was a number of readers who were interested in listening to this. So we have a streaming here as well, which is great. But I'm very, very pleased to introduce Assistant Professor Boyko from the University of Cornell. Dr. Boyko is a graduate from the University of Illinois. And he has a master's in science in computer sciences and also PhD in biology, so very skillful in that sense. After the PhD, he worked in the Stanford Medical Center as a postdoc, and then started his own laboratory in Cornell in 2011, so he's been working like 5 years now there. Using dog as a model, evolutionary model, and as a model for biomedicine as well, so you're very much welcome.
Thanks, Hannes. This is really exciting for me. This is my first time in Finland, so I really appreciate the opportunity to come here. This is probably one of the 4 or 5 big centers for dog genetics, so it's exciting for me. So my lab specializes in canine genetics, and there's several different aspects of canine genetics that I think are interesting. One of the things is that dogs are the first species that were domesticated by people, and this is so it makes them special in that way from an anthropological standpoint. And the fact is, unlike a lot of other domesticated species where we actually have a really good record and the genetic information is really clear about what happened, with dogs there's still debate about even when and where domestication happened, and of course why and how. And so that's an interesting question to ask. Additionally, there's a ton of phenotypic diversity in dogs, probably more so than most any other species, domesticated or not. So there's 2 orders of magnitude difference in adult body size in dogs. Clearly there's extreme behavioral and morphological adaptations for different breeds. To the point that scientists have asked, is there something genetically special about dogs? Are they predisposed to being able to be selected for these vast phenotypical differences? And as well as the fact that dogs have this kind of unique genetic structure that really facilitates mapping, and so it makes them potentially a very powerful genetic model species. So there's over 400 different species or breeds of dogs alive today. And again, there's a huge range of phenotypic diversity. Additionally, these breeds have very different prevalences of different inherited disorders. And so this is sort of the way that geneticists think about dogs. So the dog genome is now 10 or 11 years old, and so we see we've got dogs were domesticated from gray wolves. Something happened here at domestication. You have domestic dogs, and then in modern times, you've got different purebred closed populations that have been bred and that may have different traits and different disease propensities. And so now what the paradigm is that we use these mapping arrays, these SNP arrays, where we investigate 100,000 or so markers across the genome. And we ask the question, so for all the individuals, let's say you pick a breed and you genotype a whole bunch of individuals that have this inherited simple recessive disorder, and you look for this shared haplotype here among all the individuals from a breed that have it. And so you might get this region of 20 or 25 markers, and this is maybe 2 megabases long, and it has 20 different genes that could possibly be causing the disorder. But then you look in related breeds that also have the same disease, and you see these shared haplotypes, and you can very quickly hone in on a region that maybe just encompasses 1 or 2 genes that you can then go through and you can sequence and you can figure out what the causal variant is, and then you can, and that really is what enables you to determine how should we treat this, what sorts of therapies are there, can we predict which dogs are going to get it, can we advise to breeders about how to cross these breeds, and so we've wound up being highly successful at mapping these sorts of simple Mendelian disorders as well as traits where we have large effect loci causing it. So we've got over 200 known mutations now in OMIA, and we can start to look at the distribution of what causal variants look like. I don't know whether you can say much evolutionarily, because some of this is looking under the lamppost. It's easier to find a coding SNP variant that causes a disease, and we know lots of stuff is not caused by coding variation. But it is a great system for mapping. And so dogs are this great genetic model species, not just for disease, but also for phenotypic diversity. So we have lots of size and shape variation in people, but we have way more in dogs, and it's been positively selected. And so that makes it a lot easier to identify genetic variants underlying that diversity, because there's just more heritability and there's more large effect loci that are out there to map. And so it's this model for how do you evolve adaptive phenotypes as well as disease phenotypes. For translational research, it's terrific. So even though humans and mice last shared a common ancestor before humans and dogs did, because rodents evolved so quickly, there's actually more of the DNA shared between dogs and people. And so you're more likely to find a synthetic region in dogs that is involved in a homologous disorder to a human disorder. And then the phenotypes are more likely to be similar as well, because dogs are closer to us in body size, closer to us in longevity and the sorts of age-related disorders that they get. They share the environment with us. Some cases, they're even better than humans to look at environmental correlates. So over 90% of the dogs just drink tap water from the home, and that's the only thing that they drink. So if you're interested in what effect does water quality have on certain aspects of health, studying dogs is probably better than studying people. Also, they have a 10-fold higher aging rate, and so you can in the course of a study learn more by studying dogs than people. And as you might expect, then a lot of the homologous disorders between humans and dogs are caused by genetic variants in the same genetic pathways, oftentimes the same gene. In some cases, even the same amino acid is being mutated. So these spontaneous heritable disorders, dogs are a great system for doing it. Not that they're going to replace mice. We certainly do lots of stuff in mice that you can't do in dogs. But I think it's a great intermediate model that you can use to sort of bridge the gap between the two. And then, of course, people love dogs. And so this has a twofold advantage. So first of all, we have a subset of dogs in the world that have very high quality veterinary care. So they're getting diagnosed with the same diseases, as well as being given the same treatments as people. And so they make this natural translational model, and you don't have to house the dogs like you have to house mice. You're actually, the owners are paying for this, and you just have to figure out a way to get that information. And then, of course, they're an excellent system for education and outreach. People love dogs. People understand dogs. As a geneticist, I think about Darwin's finches when I think about origin of species, but if you go back through there's more pictures of dogs than there are finches, because it's a great way to explain to the public evolution and what's going on. Now, despite all these advantages, there's still a lot of things that we don't know about dogs. So as I said earlier, where did dogs come from? We're still trying to figure out the domestication story for dogs. We did a really good job of using dogs to identify simple Mendelian disorders, but of course, dogs also get complex diseases, and here it's a more difficult mapping problem. We don't know, for the most part, the genes that underlie different cancer risks in different breeds, or different hip dysplasia risks in different breeds, or autoimmune disease. We also, even though we know the genes that underlie fur type and coat color and body size in dogs to a very large degree, we don't know a lot about the genetic basis for canine behavior. So what would we need to do in order to figure that out? And then, of course, ultimately, we're interested in how do we breed healthier dogs. And so these are sort of four very disparate ideas, but this is kind of what I want to talk about today, because they're all related from the standpoint of we just need more data. If we have big data techniques where we can get genetic information and other sorts of clinical and phenotypic information from enough dogs, all of these become tractable problems. And so this is something my lab's been working on, as well as other labs around the world. And so the project I'm going to talk about today was a collaborative project between Cornell and Zoetis, working with Illumina, to actually do dense genotyping on a large cohort of dogs. So we have a biobank at Cornell for the animal hospital, and so we were able to look at nearly 5,000 purebred dogs from that biobank that have been diagnosed with different clinical disorders. We've added in a diversity component. So traveling around the world, we've collected over 500 village dog samples that we included in this project from 40 different countries. We improved the mapping array in dogs. So we found variants from sequencing studies that weren't well tagged by the array. We put them in We added Y chromosome and mitochondrial to get more information about this. So for the start of the talk, I want to talk about the village dogs. So people ask me, what is a village dog? There's 1 billion dogs alive in the world today, and most of them are not purebred dogs. Most of them are not pet dogs in the way that we think about pet dogs, where we control the breeding of the dog, we provide food and veterinary care and all of that. These are free breeding dogs, often free ranging dogs. These are found throughout the world, particularly in developing countries now, but they're sort of what dogs evolved to be. When they became domesticated, this is what dogs looked like, and although humans certainly bred dogs to do different roles, throughout history, most of the dogs in the world were not bred to do a certain role. We didn't control the breeding of these dogs at all. And so we call these village dogs after Raymond and Lorna Coppinger sort of coined the term and popularized it. And so village dogs to me are this intermediate link between the gray wolves that first became domesticated. But we have this large population of dogs. They diversified fairly rapidly throughout most of the world. They're basically found everywhere there's people today, except for now where we've actually have animal control that's taken them out of a lot of developing countries. And then we've, through ancient and modern breeding, they were sort of the ancestral stock for the dog breeds that we see today. And certainly we have gene flow between wolves and dogs. Certainly in many places now we see free-ranging, free-breeding dog populations where we actually have an influx of modern breed DNA into those populations. But I think it was an open question when we started the study, how many places in the world do we actually see true indigenous dog populations that haven't really, they're living in a human environment, but human breeding hasn't really controlled the numbers. So from a domestication standpoint, studying these dogs is interesting because we don't know exactly where dogs came from. We know they came from gray wolves, Eurasian gray wolves. So Native Americans had dogs too, but the signatures in those dogs match the signatures in Eurasian dogs. So we know that they came from Eurasian gray wolves and then spread out. But of course, the spread of Eurasian gray wolves is pretty strong. If you look at archaeological sites, so the more green the circle is, the older the archaeological site where we know that there's dogs. We see the earliest dogs show up, almost throughout the expanse of Eurasia. So they're not very informative for where the first dog came from. Some of this is sparseness in the archaeological record. And we can look at some breeds of dogs. So Finnish Spitzes and chows and Tibetan Mastiffs and stuff, where most dog breeds came from the Victorian time in Europe, but there's a few more ancient breeds that are still alive today. And so we can start using these breeds to infer something about the ancestry of the first dogs. But of course, these breeds are, for the most part, Basenjis, they're very spatially isolated. They've gone through bottlenecks. If you try to use most kind of statistical genetics methods, what you're seeing the loss of diversity in modern times and not any sort of signature, further back in time. So you're catching these bottlenecks here, and you're not catching this over here to figure out what happened at domestication. So in some sense, just studying the purebred dogs, which is what most dog geneticists do for all sorts of good reasons, including you can map genetic disorders that way, is probably not the best way to look at dog origins. It would be like trying to infer human origins by sequencing all the European royal families. You're not going to get the information that you're really looking for to localize that process. And so in my lab, starting when I was a postdoc with Carlos Bustamante and moving on, my brother and I have kind of gone around the world. We've sampled dogs from a whole bunch of different areas. So dog sampling in the field, as you can imagine, is a little bit of an event for the people in the villages. But we go through, we give the dog treats, we put a muzzle on it, we draw blood from the dog. They're a great species for studying in the field. So when I was a grad student, I studied tropical butterflies, and so you'd have to go out with a butterfly net at dawn and wait around until you find the right butterfly and try to stalk it and catch it. And with dogs, you just bring food and they show up, and you try to keep the puppies out of the treat bag, and it's really fun. We just draw some blood from the cephalic vein when we're there. We also rely on collaborators that are in countries that we don't go to, and they can sample saliva. We can get DNA from that. And then we can put all this on a map. So we can look at principal component analysis, and we can combine village dogs and purebred dogs. And so most of our samples that we did were purebred dogs, and you have probably most of those samples are right here, which is where all the European dogs fall. And then we see there's some, of these indigenous or these ancient breeds, Akitas, Sharpeis, Shiba Inus, Chows, that we can get along. And so we have this axis here that goes all the way through Asian diversity, and we have this axis here that goes through African diversity, and then intermediate we get all through Central, Southern Asia, places that we've sampled. And so we have this geographical clustering. So we get American dogs cluster with European dogs, which is sort of an indication that the dogs that we see wandering around the neotropics are descended from the colonist dogs and not from the Native American dogs. At least predominantly, that's a genetic signature. But we also see large regions of the PCA plot where we've got dog samples that don't really match up with any of the purebreds that we're seeing. So we are filling in a lot of the missing picture of dog diversity because these aren't in breeds that have sort of come into biobanks now that we're able to study. So they're natural populations. So we thought maybe, okay, let's compare this to wolf DNA. Can we figure out where dogs maybe were domesticated from by looking at different wolves? And so this is kind of a small diagram here, but this is a tree mix diagram. And again, we see the spatial clustering of the dogs. So these are African dogs, these are Middle Eastern dogs, these are Asian dogs. And they form a monophyletic clade, and the nearest outgroup is Middle Eastern wolf. And so we're like, oh wow, this is a great signal that Middle Eastern wolves are closely related to dogs. And so probably domestication happened in the Middle East. And so this is, so we draw this population map. We allow for admixture. So we know some African populations have European DNA in them, for instance, and likewise with some of these Pacific Island populations. But unfortunately, if we run the algorithm with a different number of migration edges, it paints a different picture, right? So if we look at 16 edges, then dogs are still monophyletic, but now we have Chinese wolves as being more closely related. And what's going on is that there's actually admixture between dogs and wolves, and these are more subtle signatures than what we're picking up on here with modern breed dogs that are admixed locally with certain indigenous dog populations. And so we know Middle Eastern admixture actually was two-way, so when dogs admixed into Middle Eastern wolves admixed into dogs, and similarly with Chinese wolves. And so here we're not painting any admixture edge with Chinese wolves. The algorithm just didn't detect it. Instead, what it did is it just clustered Chinese wolves more closely to dogs than the other wolves. But we don't think that that's a signature of dogs were domesticated necessarily in China. It could just be we're not taking into account this admixture thing. So, and we can actually prove using different genetic methods. So this is the admixture test from Reich, that we clearly do have admixture between Chinese wolves and Chinese and East Asian dog populations that wasn't accounted for in that graph. And so we wanted to use a different way that was less affected by admixture to try to look at patterns of diversity to see if we can infer where dogs came from. And so the method we decided to use was linkage disequilibrium. So this is the correlation between markers, particularly ones that are closely near each other on the genome. And the idea here is in sort of ancestral populations, you have lower levels of genetic diversity, and as you spread out from an origin, what happens is you get these founder effects that decrease the genetic diversity, increase the levels of linkage disequilibrium. And so in people, it's this very clear signature. So if you start out in Africa, you have low levels of LD, and then as you go into Eurasia, and then you go further along through Oceania and the Americas, you get higher levels of LD, almost to the point where you have this near linear relationship between short-range linkage disequilibrium and the geographical distance from East Africa, which is the origin for modern people, and sort of follows these maps going around. And people have looked at LD in dogs before, and of course the striking pattern there is all dogs have more LD than wolves, particularly short range, and then breed dogs all have really high levels of linkage disequilibrium. So it actually goes on for a megabase or more within a breed, and they're all at astoundingly different levels, and it just depends on how inbred that breed is. Whereas village dogs tend to be very consistent in the level of LD that they have. And so with our high geographical resolution, we could actually look at these patterns at very short intersnip distances. We put it on a recombination map, so we have some idea how far apart they are. And so we see this consistent decay of LD pattern, although we also consistently see some populations with higher levels of LD than others. And when we put it on a geographical map, what we see is actually the lowest levels is sort of centered here in Central Asia. So we think that this is probably where dogs originated and then spread out through several different axes. And then we had some population turnover and whatnot. We did simulation studies too to just make sure it wasn't because Central Asia is in the middle and that you have gene flow between neighboring populations. That doesn't seem to account for the patterns that we're seeing. And so, the model now that we think is going on is that you had wolves that were present in Central Asia, you had Paleolithic people that were there, they were both hunting large game, so they interacted with each other around kills, probably through climate change or maybe an increase in human population size or hunting efficiency. You had pressure on these prey populations where it became beneficial for wolves to start scavenging from human kills rather than obligately hunting. And as soon as you got to the point where they started to evolve genetic adaptations for scavenging, things like smaller body size, things like tameness, earlier age of first reproduction, you sort of set this genetic path where hunting became less and less profitable and scavenging became more and more profitable. And that's what led to this one wolf population evolving to become a dog. And it was pretty beneficial. There's 1 billion dogs in the world today. There's probably only 1 million wolves. So they've done pretty well for themselves. So I could talk about, village dogs and that for a full hour or more, but I'd like to shift gears now. Most of the dogs that we sampled were purebred dogs, and most of this study was done because we were interested in mapping complex genetic diseases in dogs. And so the 7 diseases that we had the largest number of samples from the biobank were dogs with hip dysplasia, elbow dysplasia, cranial cruciate ligament disorder, so orthopedic conditions. We also looked at a couple different cancer types. We looked at liver disease. We looked at cardiac disease. So we had 7 different complex diseases with 250 or so or more samples that passed our phenotypic criteria. And out of these 7 diseases, we actually had statistical associations with 2 of them. So 2 out of 7. And they're pretty good candidate genes. We're doing some work to follow up on both of these. Particularly, we're interested in validating them in other populations as well as doing functional characteristics. Rather than go into the specific diseases, the striking thing when I talk about this project, so I work at a vet school, and you tell the veterinarians, hey, we just did one of the largest GWAS studies ever in dogs, and we had about a 25% success rate. And they're like, oh, it's hopeless. Like, there's no way we're ever going to have studies that are big enough that we're going to be able to have the power to identify stuff. And then, talking at the New York Genome Center to a bunch of human geneticists, they're like, holy cow, you were able to map 2 diseases with just using a couple hundred samples? That's amazing. You'd never be able to do that in human genetics. So we wanted to look at this idea of power a bit more, because there's lots of genetic diseases beyond these 7 that we're interested in, and 7 is a pretty poor sample size. Maybe these were diseases that we chose that were particularly hard or particularly easy. I don't know. And so there's so I have a DVM PhD student in my lab, Michelle, and so most of the dogs that we had were patients at the Cornell University Animal Hospital, and one of the things many patients at the hospital get are blood chemistry and CBC panels that are run. And so through these we have almost 40 different phenotypes that are measured in a related, in a consistent way for all these dogs that have these blood panels run. And the traits themselves are interesting from a disease standpoint. Many of these are actually easier perhaps than diseases that may be heterogeneous. You can actually identify subparts of the disease, and so they could be related to clinical conditions. And so over 1,000 of the dogs had these blood panels run. Unfortunately, a lot of the dogs are undergoing chemotherapy or liver failure or something like that that's really going to bias the measurements. But we set up this phenotypic criteria. We excluded dogs with certain diagnoses or dogs that otherwise were outside of the range that we're normally encountering. And so we had 353 dogs that were deemed healthy enough. And so we could do GWAS on 39 traits using a sample size of 353. So very similar to what we set up with the actual diseases. And what we found is for 39 out of the 39 phenotypes, 9 of them had significant genetic associations. So almost the same proportion, about 1/4 of the complex traits that we looked at with this sample size had associations. And some of the associations were I thought, quite interesting. So this was our positive control. So there is a known copy number variant in the amylase locus. So dogs have, like people, they have different copy number, different copy numbers of amylase gene, and this correlates to starch digestion and the amount of circulating amylase in the blood. And so even though we don't have the actual copy number on the array, we do have enough LD with it that we could identify a very significant association with the right locus. But we also found this novel association, very strong, for alanine aminotransferase on chromosome 13. And what's interesting is it's right over the GPT gene, which is the gene that actually encodes the transferase. So we have a very good candidate gene. We haven't done the mapping yet to find out what the causal variant is. But if you look at this haplotype, you see that so healthy dogs are right here in the center of the reference interval. But as you get increasing numbers of copies of the mutated haplotype, your ALT levels in the blood go down. And so it looks like it's behaving in an additive sort of way. And for us, what was most interesting is, so ALT is this measure for liver disease. And so if you have a high level of ALT, then the vet is on the lookout for liver problems and it will diagnose you with liver disease. But if you actually look at the dogs that are diagnosed with liver disease, we see that we see the same sort of pattern. So we're getting a reduction, and so on average, a dog with liver disease so this is the 100 is sort of the limit that we use for the reference interval. You have above 100, you've got elevated ALT, and so that's sort of a signature. And so the average is above it, but the mode is still below 100. And so we think there's this potential to misdiagnose a dog if you're just using the same reference range for all of the different dogs. So in humans, you don't ever see a locus that has this large of an effect on a single blood phenotype, certainly not at a high frequency. And so this sort of points to the idea that in dogs, we do see large effect loci, and they actually could contribute to how we diagnose dogs clinically. So it sort of points to the importance of doing genetic testing. So why are we only seeing a 25% power in our study? We did a bunch of simulation analysis. One thing our study did different than other studies is we looked across breeds. And so when you first start doing mapping studies, the canine HD array was designed to do cases and controls within a breed. That's sort of the paradigm that's been used by mapping. And the reason for that is clear. If you go across breeds, you actually zap yourself with a lot of power because you have to correct for population structure, and that correction is going to reduce power. But that's not the only thing that reduces your power. So if you've got low numbers of samples, you're clearly better off if you can get all those samples from one breed. But the advantage is if you look across breeds and you have access to a biobank, let's say, if you have enough money, you can certainly find more samples if you're allowed to collect from multiple breeds than if you're only able to collect from one breed. And for complex traits, this is particularly important because we're interested not just in why some golden retrievers get cancer and other golden retrievers don't, but we're also interested in why do golden retrievers seem to get more cancer than other kinds of retrievers. And so if we want to look at those genetic loci that are leading to those differences, we really want to include as many different breeds in our study design as possible. And our power increases, of course, as we get more and more samples. But beyond that, the reason why we started out with such low power was not just because we had to correct for the population structure, but it's also because, thinking back to the linkage disequilibrium, within a breed you have very long range LD. So causal loci tend to be tagged very well by 100,000, 200,000 SNPs. But when you're looking across breeds, if you had a denser marker panel, you can actually improve your power still further. So the idea is we need not only larger sample sizes, although maybe not prohibitively large, maybe not to the tens of thousands like we have in people, maybe 1,000 cases and 1,000 controls is actually going to give you 80% power to map many complex diseases, depending on the genetic architecture and how much of an effect the causal loci that you're looking for have. So if you're looking, if you're trying to map something with an effect size of half a standard deviation or one standard deviation, right now if you do a cross-breed study with 500 cases and 500 controls, you really have no power here and you have moderate power here. And you can increase that either by doubling your number of samples or by improving your marker density for the array that you're looking at. And ideally you do both, and then you can have appreciable power to detect those sorts of moderate to strong effect loci for complex disease. So another complex trait in dogs is behavior. We know there's a lot of behavioral variability in dogs. dogs have been bred to perform many different roles. So I think part of the reason why there's so much phenotypic and behavioral diversity in dogs, unlike, say, domestic cats, is that dogs can be motivated. And so you can get the dog to do something. And so therefore, we've bred dogs to do a whole bunch of different stuff. And almost anything you can think of, a dog has been bred for that. And so that involves not only changing how big the dog is or what the shape of the dog is, but also how the dog behaves. And a way geneticists think about adaptive evolution is through positive selection, which often occurs through selective sweeps. So the idea is you have a new variant, or you have a variant that maybe was in the population, but now the environment has changed, or the selective forces have changed, and so that variant is adaptive. And so it rises in frequency, carrying a haplotype along with it, and we call this a selective sweep. And these are things that we see in humans. So we see selective sweeps for hair and pigmentation genes. We see selective sweeps for disease resistance genes, particularly things involved with malarial resistance. So we'll see high frequencies in malarial endemic regions, and then low frequencies are absent elsewhere. And then we also see for metabolic sorts of traits, lactase persistence and alcohol dehydrogenase and amylase copy number variation. We see sweeps for these sorts of things in people, and lots of differentiation between different populations. If you look for the same patterns between dogs and wolves, you can identify sweeps that seem to be associated with dog domestication or shortly thereafter. And these are things, more than half of them seem to be associated with behavioral or neurological, developmental sorts of pathways. And then we have other things, particularly starch and fat metabolism, including amylase copy number variation. So dogs don't have salivary amylase. They have pancreatic amylase, but otherwise it's sort of the same situation that you see in people, where you see this increase in copy number where starch digestion is important. And so we were interested in looking across the breeds, where we see lots of morphological diversity, lots of behavioral diversity, and see what selective sweeps in dogs look like across breeds. This has been done before, and so we know we can expect coat color and fur type loci, body size loci, particular traits, dog shape and whatnot, to show signatures of selective sweeps. In fact, selective sweep mapping in dogs is so nice that in some cases we didn't know what trait we were looking for. We just saw that there was a signature of positive selection, and then we could figure out that, oh, so this is the gene for wrinkled skin, or this is the gene for droppy ears or curly tails. But doing all of this, so far nobody has really found compelling evidence for a selective sweep for behavior. So we have 5,000 dogs from 165 different breeds that have been genotyped, and so we thought, well, maybe we can apply our data set and maybe correlate it with other data sets and see if we can actually see if there's any evidence for positive selection sweeps for behavior. And so the method we chose to look for positive selection in dogs is called HapFLK. So this takes haplotype information, not just SNP information, and it takes the underlying structure, like phylogenetic structure of the different breeds, and then tries to find regions where the haplotype frequencies differ based on what you would have expected for the overall genome-wide sharing of haplotypes across the breeds. And so you get this genome-wide normal distribution, and then you get this observed distribution. You have this fat tail on the right-hand side, and these are putative regions of positive selection. So we sort of set a cutoff. We have 68 highly elevated regions, and as you go along the genome, looking at these positive selection scores, you see they occur in these particular peaks, and many of these peaks have already been associated with things like body size, or skull shape, or morphological traits. But we also found regions that hadn't been associated with anything yet. So this is chromosome 1, this is chromosome 3. The chromosome 1 was actually really interesting to us. So I had a graduate student that was looking at these positive selection scans, and then I had a postdoc, Jess Hayward, that had been looking at doing association mapping. And this strong signature of positive selection on chromosome 1 actually matched perfectly with this QTL that she found. So it's right here in these this pair of melanocortin receptor genes. And in fact, the strongest signal out of all of the markers in this region on the array was a missense mutation in MC5R that we had put on the array specifically because we saw it was segregating in village dogs and we didn't know what it did. And when we did a GWAS study, what we found is that for fur type, we found that this is related to fur length, and specifically, it's most related to how much shedding of the coat that the dog has. And so in dogs, it looks like fur shedding is primarily controlled by 3 genes. So you have this respondent 2 genotype that gives you the furnishing look. And so if you have a poodle type coat, then you're going to be a low shedding breed. But if you have a regular sort of non-furnished coat, then the determination of whether you're sort of a heavy shedding breed versus a lighter or average shedding breed depends on whether you get this short boxer-like, pug-like coat or this somewhat longer Akita-like, wolf-like coat that's going to shed a lot more. So it was kind of fun to see this overlap between positive selection and this new trait that we mapped. So we can take a genome-wide view and we can look at all the different regions that we found evidence for positive selection that had already been associated with morphological, like body size, body shape variation, as well as coat type and fur color variation. We also saw evidence of positive selection in the dog MHC region. But then we also saw a couple dozen regions that show evidence of positive selection and so far have not been associated with a phenotype. So we had this underlying hypothesis that if there's selective sweeps for behavior, then they're probably going to be in one of these regions in the genome, hopefully more. So the problem is we didn't have behavioral trials that were run on the dogs that we had genotyped. And so what we were hoping is that there's enough behavioral variation across breeds that we can say, okay, breeds that exhibit this behavior have these allele frequencies, and breeds that exhibit this behavior have those allele frequencies. And so to get a large data set of a whole bunch of different canine behaviors, we collaborated with James Serpell at Penn, and we took his CBARC data set, this research questionnaire with 100 questions that's been asked for tens of thousands of owners. And for the 108 breeds that we had in our study and that he had in his study, we wanted to see if we could find any correlations between the behavioral scores for those different breeds and the allele frequencies that we had seen in our study and that were used for determining the regions of positive selection. And as you might expect, we had lots of associations come out. And again, some of these regions do seem to overlap regions that have been found in other studies. And so we're trying to do follow-up. There's good candidate genes for some of these. But we wanted to see what the overlap was with our positive selection scan. Do we see, so this is the genome-wide distribution, right, of all the markers on the array. And if you looked at markers that are associated with body size that we found, you can see it's shifted over to the left here. So things that are associated with body size tend to be positively selected, some of them very strongly so. So for our top hits for all the behavioral QTLs, did we see the same excess? And we did somewhat, not to the same extent. So we saw lots of stuff that didn't really look selected. And so, maybe this is a behavior that wasn't positively selected, or maybe just a spurious association, which can happen when you're doing breed mapping sort of stuff. But we do see this subset now of variants that do look like they're positively selected. And so my grad student, who was very interested in behavior and finding genes for behavior and all that, excitedly tried to match up everything that was going on. And unfortunately for her, what we were finding is we would see like nervousness on stairs had positive selection, but that was for the gene for short legs. So it wasn't really a behavioral gene. And dogs with large bushy eyebrows tend to chase shadows and snap at invisible flies. And dogs with floppy ears are rated as more fearful of other dogs by their owners. And so these are things that either the morphology probably directly impacts the behavior, or it impacts the owner's assessment of the behavior, and it's not that it's a separate kind of brain gene or behavior gene that's driving it. And so sort of as a last straw, what we did was we excluded everything that we had previously associated with a morphological thing. So I think in some cases it's possible that you have two different linked phenotypes, and so maybe there is still something for behavior in some of these haplotypes. But if we just look at things that have not been associated with the morphological thing, we're down now to two different questions, two different QTL signals, and the out of the 100, and the two were excessive drooling when left alone and howling when left alone. And these were certainly not the two traits I had thought we were going to be mapping. When I started out on this study, we were looking at things like aggression and trainability and all that kind of cool stuff. But something's better than nothing. So we looked at excessive drooling when left alone, and we find this signal in chromosome 5. And then when we looked at the breeds that were driving this signal, we very quickly realized that what we should be doing is the GWAS for, if you look at the breed standard, whether or not it says the breed has pendulous flews or hanging lips. And in fact, then you have a really, really strong association. So really what we found is that we had a strong signature selection for large floppy lips, and that leads to drooling. So we actually, again, we mapped morphology, except we did it intermediately using behavior. And so for the chromosome 12 locus, we sort of decided to just use Google image search to figure out what was going on. And sure enough, the dogs that howl a lot seem to have especially big ears. And there's actually a growth factor that is being mapped here. And so we're trying to do some follow-up work, because I'd be very interested to know, is it actually the growth factor? There's several genes in the region, and is it impacting ear growth and like the larynx in some way? Or is it just that dogs with large ears hear the world differently and maybe vocalize differently? Like, we don't really have a clear answer on that. But I think that the conservative hypothesis here is that the selection was for the ear type and not for the howling behavior. And so overall, we're not seeing sweeps, positive selective sweeps, when we do breed mapping for behavior. So this suggests two things. First of all, the genetic architecture for behavior is likely very different than the genetic architecture for morphological variation. And then also there's probably a lot more variability in behavior within a breed than you see for some of the morphological phenotypes. So really we had an underpowered study. What you need to do is you need to get lots of dogs genotyped with consistent behavioral assays done, ideally in a consistent environment, because we know some of this is heritable, it's out there, but maybe it's not structured in terms of positive selection sweeps. And so anyway, in the time I've got left, I would like to talk a little bit about breeding healthier dogs. And so basically all I'm going to talk about right now is inbreeding, because that seems to be like the big thing people care about. Obviously, purebred dogs are inbred to some extent, more or less. So this is your typical Labrador Retriever. It's about 20% inbred, and so it gets these large tracts of homozygosity throughout the region. And this dog here just has one chromosome 23. It inherited the same copy from mom and dad. And village dogs don't have that. These inbreeding tracts are interesting for two reasons. First of all, you can overlay all the inbreeding tracts from individuals from a breed, and so you can identify regions of the genome where, there's basically one high-frequency haplotype in all the Labradors. So something here underlies Labradorness, but it might have also swept to it deleterious alleles too that might be important for disease. So I think doing these sorts of inbreeding maps are interesting. They're different for different breeds, and we can also see the effect of population size and effective population size within breeds. So it's not like Irish Wolfhounds that had this huge degree of positive selection, and there's 100 different regions of the genome that got selected for. It's that there was a lot of drift that happened to fix in a lot of different regions. And so maybe that has a genetic consequence for the breed. So fortunately or unfortunately, depending on your viewpoint, most of the dogs in our study are still alive. So we don't have information about like longevity and stuff like that from the dogs that we looked at. But we can go through. There are published databases of dog genetic information. So we just built a web crawler. We went to caninedna.com, and there's over 10,000 golden retrievers that have date of birth, date of death, 10-generation inbreeding coefficient, all this sort of information there. And we can see whether or not longevity data from these records make sense. And what we see is this pattern of females live longer than males do, which is something that I think makes sense for dogs. We see a pattern that golden retrievers in the UK live longer than golden retrievers in North America do, which would also kind of agree with the fact that we have a subset of diversity that has made it over into North America, because golden retrievers came from Europe. And then we also found that after you account for these effects, if you look at the 10-generation coefficient of inbreeding in these golden retrievers, basically every % increase in inbreeding decreases the expected longevity by a month. So we can see, just based on longevity data alone, that inbreeding is deleterious. And we can see there's lots of different ranges of inbreeding, both within breeds. So these are our golden retrievers, and we have some golden retrievers here. So this is now based on the SNP data, and just estimating the inbreeding tracts from that. Some golden retrievers have less than 20% inbreeding, some golden retrievers have 40% inbreeding, and the mean is about 20%, and the means for all these different breeds are very different, whereas for the village dogs, it's basically all zero unless unless you're collecting dogs from an isolated island. And so we have size data from our dogs. We know which breed our dogs come from. And so we can build linear models, and we can actually see, okay, well, sex is a significant effect of how big the dog is. But inbreeding is also a significant effect of how big the dog is. So the dogs that we see that are inbred for their breed tend to be small for their breed as well, which is consistent with we have deleterious recessive variation that gets unmasked that reduces growth and also reduces lifespan in the dogs. Now, we worked with AKC to get access to pedigree records from the dogs and also look at the number of registrations we had in the US, which is where our dogs came from. And surprisingly, the average level of inbreeding within a breed did not correlate at all to the number of annual registrations that we had in the breed. So this inbreeding doesn't seem to be related to the actual census population size of the breed, the changes in the mean inbreeding. So we think it's more historical patterns. But beyond the historical patterns, just like how big was the bottleneck at the founding of the breed, we did see a correlation with the average body weight of the breed. So larger breeds tended to have more inbreeding than smaller breeds did. And it was a highly significant effect. And in fact, when we parsed it out by looking at Y versus mitochondrial diversity and looking at haplotype diversity on X versus autosomes, we could see that that pattern was driven by the fact that big breeds tend to have small female effective population sizes. So pretty much all dog breeds have small male effective population sizes because of popular sire effects. But I think for the larger breeds, you have fewer litters per female, and the litters that they have tend to be much larger. And so I think you just have fewer breeding females overall. I think it's, and also it becomes difficult for a typical owner. I mean, you can probably place 4 to 5 puppies, but you're not going to place 10 to 12 puppies. You kind of have to be more of a professional. So I think it's both this ecological as well as this socioeconomic thing going on, that you just have smaller female effective population sizes in these big breeds. And so we see this pattern now in overall effective population size for those breeds. And of course, big breeds tend to not live as long as small breeds do, which makes dogs interesting in and of themselves. And so the natural question is it because the big breeds are more inbred, or is it size per se that's driving it? And so we could do a linear model on that too. And in fact, once you account for size, the inbreeding level goes on either side of the diagonal pretty easily. And so there's really strong effect of size and basically no effect for inbreeding when you're looking at mean inbreeding across populations. And so the conclusion from that is that if you're looking within a breed, the differences in the inbreeding levels in individuals are primarily driven by inbreeding from the last couple of generations. And when you're looking at across breeds, you're looking at things that happened much, much more anciently in time. And so it's stuff that's had a chance to kind of, if there was a strong deleterious mutation, get weeded out. And so it's unlikely that you have highly deleterious variants now from these older haplotypes sharing inbreeding, whereas more recent in your pedigree inbreeding, there's more likely a chance that you have a big deleterious effect. So recapping, from our data, it looks like the highest levels of diversity are in Central Asia, making this a likely origin for dogs. We think that complex disease mapping in dogs is not hopeless. We just need to get good phenotyping, we need to get enough samples, and we need to get better tools for doing it. We think breed mapping works great for size and other sorts of traits that are more fixed within breeds. For behavior, it doesn't seem to be working as well, and so we need other ways to go about doing that. And also, not all inbreeding is created equal. So more ancient inbreeding seems to be less harmful than more recent inbreeding does for dogs. So going forward, some of the stuff my lab is working on is just improved resolution of genotyping platforms. So we've resequenced now 295 dogs total, including publicly available dog sequences, and we've used this sequencing data now to develop with Illumina this new 228,000 marker array, which fills in a lot of gaps that we have in the genome. And so now we can double this array with the sequencing data to actually do imputation. And so we're working on this imputation so that if you run a large number of dogs in a genotyping setting, instead of getting 200,000 markers out of that, you can get 10 million markers through imputation from the reference panel. And we're also trying to improve variant identification and annotation, and we're doing this primarily through projects to look at eQTLs and methylation QTLs, and also for cataloging structural and copy number variation in the dog genome, which also seem to be important. And then finally, continuing to work with village dogs so we can look at not just the mutations in purebred dogs and identify them, but then to go back and see what was the evolutionary trajectory of these. Where did they arise? When did they become selected for? And all of that. Also along these lines, but also Cornell mandates, conflict of interest disclosure. So I'm a co-founder and officer of Embark Veterinary. So we're trying to use consumer canine genomics to also improve scientific discovery. And with that, I think I'm done. This has been obviously a huge collaboration among many people, both at Cornell and elsewhere. I started it in Carlos's lab. We've gotten funding from diverse sources, including National Geographic, NIH, NSF, crowdsourcing through petridish.org. And with that, are there any questions? Thanks.
Thank you very much.
Thanks.
I think we have time for a few questions. So. Yes.
Thank you for an interesting thing. I was waiting and waiting. When do you say that the big dogs live shorter, and then just in the end?
That's right, yeah. It's about a twofold difference in mean lifespan between the big dogs and the small dogs, which is much more than you see.
So it's not the myth, it's the truth.
Yeah, no, it is absolutely true. So that's the primary difference. We also see that mixed breed dogs seem to live about a year or so longer than a purebred dog of the same body size.
Yeah.
So yeah, there's lots of...
My question was, in the human area, we have tremendous problems to find genes which are linked to mental disorders, psychiatric disorders, etc., etc. Where are you going in that direction with the dogs?
So the hope, and I actually was talking with Hans about this earlier today. So yeah, I mean, the hope is that we can start to identify genes associated with behavior in other sorts of mental disorders and whatnot in dog. And I think it's a tremendous opportunity as a model organism. So I have a whole thing on mapping body size in dogs and how much easier it is because we've selected for really diverse sizes. And so you have genes of large effect that are segregating at high frequency, and so you can identify much more easy than you can in human studies. And I think the same is true for behavior. You just have to phenotype the behavior properly, but clearly there's been strong selection for very different behavioral profiles. And so we should be able to get at the genes of things even involved in mental disorders in dogs much more easily than we can in people. And so I think it's a great model species for that.
Do you select then the village dogs or breed dogs?
I would select the breed dogs. So I think the village dogs, there's still enough natural selection going on. I mean, they're really interesting, but you need a very dense marker array if you're going to get at that. And it's also, I've done some, like, with a village dog, like pointing at a cup to see if it can figure out where the food is kind of stuff. And it's kind of hard to do those experiments on a grand scale. But again, so we don't do size mapping in the village dogs either. Like the genes we find in the purebred dogs are segregating in the village dogs, and they do cause differences in size. But there's a lot of small effect loci for size, like in human populations, that are in the village dogs, underlying local adaptation and whatnot. And I think the same thing's going to be true in behavior. So you really want to look at the purebred populations where you've simplified the genetic architecture, because you just have less variation segregating to begin with, and where you've sort of fixed or raised to high frequency certain variants that are selected in some breeds and not others. And let's try to find those variants first. So that's where I would start the study.
Hi, thanks for the nice talk. I go along this question. The problem in mice, for example, is that they don't really have those human diseases, for example, autism or schizophrenia. So how much dogs are having, for example, autism or schizophrenia, or how related they are to the human diseases?
Yeah, nobody knows that yet. And my hypothesis is there's probably a lot of it, and we just don't know what an autistic dog looks like. Like we just don't know what phenotype we should use to identify it. But in humans, there's a huge mutational space. Lots of different genes contribute to autism. They seem to be highly penetrant mutations, but they're very, very rare because they're selected against. If anything in dogs, you've relaxed the selection, at least in some breeds, probably pretty significantly. I mean, dogs, they don't have to find a mate. We find the mate for them. So, as long as the dog is compatible with living in a home still, then it's going to get bred. And so I think the mutational space is probably just as big. So you probably have some breeds where you have pretty high frequency, at least much more so than people, genes that would make a very good autism model. So there is a lot of potential, but it needs to be a collaboration between geneticists and behaviorists, I think, to identify that.
A little bit more technical question. So when you're actually looking at the positive selection using the haplotype mapping approaches, how do you actually distinct the region of selection and genetic drift?
So that's why, we compare the distribution of the haplotype frequencies to the expected distribution based on the haplotype sharing across the breeds. So basically you build a tree, and then you look at the frequencies and see how discordant that is in that region from the tree. So that sort of drift should be acting genome-wide, whereas selection is going to be specific around certain loci. So before, we used FST scans, so we're just looking at how much differentiation there was overall across all the breeds. And the better way to do it with HapFLK, the reason why it's better, is that if you have two closely related breeds across most of the genome, but then they're highly divergent in one region of the genome, that's actually a much clearer indication that we had selection there than if you had two distantly related breeds that happen to be very divergent in a region. And so by looking pairwise across 78 different pairs of breeds, I think that controls for the drift problem pretty well.
So if you think the current, 200,000 markers is enough to pinpoint the haplotypes?
Within purebred dogs, I think it certainly is. You do better across breeds if you can impute or you have a denser panel, and so that's sort of it's a cost-benefit analysis right now. But we've implemented the imputation reference panel, and we're working on writing that up, and it seems to, at least for size variance, very well pick up on what the causal variant is.
Yes.
Your linkage disequilibrium data suggested that the domestication has taken place in Central Asia. Does your analysis allow to estimate exactly when this might have happened?
I kind of hesitate to put an age on it because we were doing chip data, and so there's an ascertainment bias to the markers that you put on there. And fortunately, the LD data is consistent regardless. So of course we have the regular canine HD chip was all boxer, poodle, pretty much. So basically European dog ascertained. And then we threw out another 12,000 markers that were ascertained in diverse village dogs. And we get the same qualitative patterns of LD, with Central Asia being the center for both of them. But methods to identify the age of an origin, they're much better if you use resequence data where you don't have an ascertainment bias, where you're just looking at the amount of heterozygosity and genetic diversity and sharing in an unbiased way. So because of that, in our analysis, we didn't do that. It does appear from several different sequencing studies that it looks like the divergence between dogs and wolves was 30,000 years ago, which is much older than the archaeological evidence suggests. And to me, that's suggestive, and also the fact that dogs and wolves tend to be reciprocally monophyletic, is that we had an extinct wolf population that turned into dogs. It's not that we have like a sister population to dogs that's still alive today in the wolves. And that separation from that wolf population and the other wolf populations was about 30,000 years ago.
One more question.
Excuse me, there's another lecture starting here at 1.
Sorry.
Okay, thank you very much.
Thanks.